Okay, so in today's lab, that is exercise 2 of lesson 5, I am going to show you the gram stain. So you all know that gram stain is a differential stain, as you have studied in your lecture, that differentiates two types of bacteria on the basis of the thickness of the peptidoglycan layer. So gram-positive bacteria has a very thick peptidoglycan layer, and it is about 20 to 80 nanometer in thickness, and the gram-negative cell wall, that peptidoglycan layer of the cell wall is about 2 to 20 nanometer thick. So the principle of the differential stain, this gram stain, is that due to the thickness of the peptidoglycan layer, the gram-positive bacteria can actually retain the color of the primary stain. So our primary stain here, the crystal violet is present. Then we have the grams iodine, which will act as a mordant, kind of a glue that will actually make a complex with crystal violet and will fix that crystal violet stain in the cell wall peptidoglycan layer. Next one is the decolorizer we are going to use, that is the gram stain decolorizer, that is alcohol. And the next one or is the secondary stain or the counter stain, which is safranin. So with all these stains, I am now ready to do my work. So let me light the candle and get ready with that. So I have just lighted the candle. Now from the last lab, I hope you have made the observations that how your broth culture looks like. So you see how turbid it is, which I actually took from trash can. You can see yours, which you have taken from your skin. So at the bottom also there is some flocculent growth. You can see a thickness, like a kind of a pellet like thing. So that growth is over here. Now let me show you for the yeast. The yeast is also, we can see a little bit growth at the bottom. That is also kind of a flocculent growth. So we will use this culture for today's stain. So let me show you one and then you can do the other one. So I have taken one new slide and labeled it that I will take the culture from the trash can and I will stain it. So this time also I'm not drawing any circle or anything. I will try to spread it as much as it is possible. So I'm taking my culture that is from trash can and taking the inoculation loop, and which I have disinfected with alcohol, and let me get rid of the excess alcohol and make it air dry. Okay, I have to make sure that it is dry, otherwise it will kill the microorganisms. Okay, so this is aseptic technique. That means I have to apply my aseptic technique to take the culture because I want to keep the culture for future use. So what is that? Watch it carefully. I'm opening the mouth of the tube in the front of the flame and creating a convection current so that no microbe from outside can go inside or any microbe from inside cannot come outside. Okay, then I am going down and trying to take out with the loop, and I am closing in front of the flame again and keeping it back. Okay, now I am, you see inside the loop the liquid is there which contains all my stuff. So I'm trying to get rid of the stuff. And I'm trying to spread out. So almost all liquid from the loop has come out. Okay, and I'm spreading it out so that I can make a thin smear. Okay, I don't want thick smear. It is not good for the microscope observation. Okay, now this will go for disinfection, and I will take the slide. If you have a cloth pin, you can use that. I'm using this one. Hopefully this will work. So I'm trying to hold the slide at the end where my writing is and trying to air dry. And it is already dry. Still I'm moving a little bit to make it air dry. So once it is air dry, now I will apply the heat of the flame from the back of the slide. Can you see? My writing is at the top, and from the back of the slide I'm putting on the flame, not very near, not very far, and I will move on to fix the bacterial cells to the glass of the slide. If I don't use the heat for the fixation, then the bacterial cells will wash off. They will wash with all the stain and alcohol I'll be using in the gram stain. Let me go a couple of more seconds and then it will be done, because this candle flame is not that strong. And okay, I'm making sure that it is properly done. Make sure that it is not overheat, but I'm doing for a little bit of more time because it is the candle flame. Okay, so now it is done totally, and now I will do the staining method. Let it cool down a little bit. Let me put it in the staining tray over here. So I'm putting it over here. Now the first step of the staining method is the application of the primary stain. So our primary stain is crystal violet. Okay, that is gram stain number 1. So I'll apply the crystal violet in such a way that it covers the smear properly. We say flood it with the crystal violet. So make sure that rest of the crystal violet stay, because you have to do another slide. So I tell my students that apply crystal violet lavishly so that it covers the slide totally, and that will be done for 1 minute, and then I'll wash it with water, and then I'll apply grams iodine. So I'll be back after 1 minute. So I came back, and now I will wash this crystal violet stain with water. So let me wash it like this. I use the dropper which is found in the kit. Yeah. So once it is done, I have to get rid of the stain. So let me go and get rid of that. I'm coming back soon. I'm coming back. Okay, now when you are draining of the stain, it should be with the running water at the middle of the sink. You should do it, otherwise it will stain. Now I will apply the grams iodine, you see, number 2. So that will also cover the whole staining area, you see, and it will be also for a minute. Then I'll come back. Okay, so 1 minute has passed and now I will wash with alcohol. This decolorization or decolorizing is very important. You can see the gram stain decolorizer. So you have to decolorize drop by drop, and it should be not more than 7.5 seconds for me, because see how I am dropping. Okay. Now I will quickly apply the water to stop the decolorization. If I do not apply the water quickly, then the decolorization will go on and all the color will be decolorized. All cells will lose their color, whether gram negative or gram positive. There will be false negative, false positive results. Okay, I'm getting rid of the stain in the sink. Coming back. Now I will apply the last counter stain, that is safranin or the secondary stain, and that will be for 30 to 45 seconds. Okay, so after that I'm coming back. Okay, so 45 seconds for me, it is 45 seconds. So 45 seconds over, you can do for 30 seconds. Now I will wash the slide with water again. Okay, so I will hold the slide in a slanting position and wash it. Okay. So I got rid of the excess water by touching the side of the tray. Now can you see the staining? Okay, so you can see how the slides are stained. When you will see in the microscope, we can see for some people it is purple a little bit, for some people it is reddish, but it doesn't mean anything. If I'm getting reddish over here, it doesn't mean that my cells are gram negative, because in the microscope, until and unless I see in oil, I cannot say that what it is actually. Even in like high power, it is very difficult to say sometimes, but it is very good or it is ideal always that you see in 100x with oil. That means with oil immersion lens you can see properly. So my slide looks like this. Can you see a little bit of purple over here and then reddish over here? So this is all about the gram stain. Okay,